Hey everyone, and welcome to the NanoReach podcast. Our guest today is NRG's own Marianne. So, for those in our audience who don't know about Marianne, she is one of NRG's own PhD students. In fact, she has finished writing her PhD thesis and is going to defend it later on this year. So, Marianne, how are you feeling about that?
I am actually very, very relaxed, probably the most relaxed I've ever been.
It's great to
Yeah,
hear.
and I'm also quite happy that I completed the work that I set out to do more than 5 years ago and that I managed to do it. And also excited to basically start a new chapter, as in like to go back to working in the industry and hopefully like contribute more than what I did earlier from my PhD experiences. Yeah.
All right. So, Marianne, why don't you tell our audience over here about the research that you did during your PhD?
Yeah, sure. My PhD project was based on improving the upstream process, that is the cell culture process via feeding strategies in order to improve monoclonal antibody production. So this was given to me with the objective of basically addressing the growing demand of biopharmaceutical as therapeutics nowadays. So from my perspective, this was a very new domain for me since I had no upstream processing experience before coming to ICT. I came from an analytical background. And also, I was the first in our lab to work in this particular domain. So there wasn't any groundwork for me to build on. It took a while to get things up and running and for me to figure out how to achieve my objectives. So a lot of the first 1 or 2 years basically just went in reading up a lot since a lot of it was new to me. And that's when I realized that the objective or other topic that was given to me was could be achieved better if I understand the consumption and accumulation of nutrients in the medium, since my project was based on feeding strategies, basically how you improve the fed batch process in order to improve antibody production. So in order to improve the feeding strategies, you need to first understand how the nutrients in the medium are being consumed or accumulated. So for this purpose, I needed a good spent media analysis method. And I also realized that probably using statistical approach would be beneficial because I realized that any kind of a cell culture process gives you so much of data that you may not always understand, say, correlations or important outcomes just by looking at the data. You probably need some amount of processing and which gives you, like correlations that wouldn't be usually visualized. So I started off with basically establishing collaborations with Shimadzu for the LCMS analysis for spent media. And I also worked with Sartorius for the multivariate statistical analysis. And so the first part of my project was basically to identify correlations between nutrients and metabolites in the culture with process outcomes such as cell density or antibody production or CQAs. So using the statistical approach, we identified correlations and therefore targets which could be used later on for optimizing the feeding process. So that was the first part of my project. Then the second was basically actually doing the feed optimization part of it. So in this part, I optimized a feed composition, or rather, I showed a feed enrichment strategy using TCA cycle intermediates. So TCA is the tricarboxylic acid cycle or the Krebs cycle. So what I showed was if we introduce these intermediates and precursors of the TCA cycle in the feed, we were able to increase MAP production by up to 55%, which was quite substantial. And in the final part of my project, I decided to explore a slightly different approach. So I had read that an approach called process intensification was relatively new, and also it was showing better increases in antibody production than traditional approaches. So one thing I forgot to mention was that my entire project was done at the shake flask level. So this is typically not the most preferred method. It's not only small scale, but it it does not have a lot of control So it's preferred that you perform cell culture experiments in bioreactors, which have better operational controls of parameters like pH and oxygen, etc. Since at the time in our lab we didn't have bioreactors, I was working on shake flasks entirely. I kind of tried to use this to my advantage and demonstrate intensification strategies in shake flasks, since this was not actually shown till now. So I did that, and I got up to 150% increase in antibody, which was not only the highest in my project, but I genuinely didn't expect that to work, especially since I'm working in shake flasks. And like I said, there's a lot of lack of control involved. So through this final study, we proposed that intensification approaches can actually be the best approach for achieving more significant increases in titer. And since we demonstrated it in shake flasks, we further proposed that intensification could be adopted in the very initial stages of process development in the industry as well, since these initial stages are typically performed in shake flasks, and since we demonstrated it in shake flasks, they can hence be adopted from the very early stages of development, and and then optimized in conventional ways. So yeah, that's pretty much a gist of my journey. And I was able to pull out 3 publications from this entire work, 2 of which are published, and the last one is under submission.
It's amazing, Marianne. I know how hard it is to get a paper published. Congrats. So you mentioned that you had worked on process intensification. Would you potentially, now that you're done with your PhD, hope to actually work on this in the industry wherever you go?
Yeah, I do. So, like I said, from my reading, I found that it's a very useful approach. I believe people would be using it. I'm generally not sure because, like I said, I was not from the upstream domain, so I have to still figure out how much it is actually being used. But it's very interesting. It's a very successful approach, and I hope I do get to use it in the industry as well.
I also had another couple of questions about the cells that you were using to produce your antibodies. So, which cells did you use?
I was using Chinese hamster ovary cell line. It was genetically modified to produce a monoclonal antibody for therapeutic use. But in my particular project, we were not focused on the antibody itself. It was more like a model cell line, since my project was more process oriented and not really making a product, so to speak. Yeah, so it was basically a model cell line and a model antibody.
And the feed that you mentioned for the cells, what does it normally consist of?
Okay, so you have in a fed batch process, you have a growth medium and a feed medium. The growth medium is the medium in which the cells are inoculated, and then the feed medium is what you supply to the culture intermittently, like at regular intervals. Both these media are almost essentially the same composition. Typically, the feed medium is considered to be a more concentrated version of the growth medium. When I say the composition is the same, in the sense that it constitutes amino acids, vitamins, lipids, trace metals, salts, etc. So everything that's required for a mammalian cell to survive, grow, and multiply.
I was reading about you and the work you had done before you came to speak with me today. You were with the industry before you started working on your PhD, correct?
Yes, I was.
Yeah. So, can you tell me anything about your experience while you were working in the industry?
Yeah, sure. So before starting PhD, I worked for a total of 3 years in the biopharma industry. First at Lupin Biotech in Pune, that was for around 2 years, and then at Acutus Biologics in Mumbai for around a year, both in analytical departments. So my experience over there was primarily based on characterizing the in-house products at the in-process level and even the final DS, TP, and stability samples. So, but all in all, it was purely analytical. I also gained some experience in method validation, method transfer, also some experience in analyzing clinical samples.
While you were in the industry, did you manage to build a professional network?
I did. I would still honestly say that my network improved here at ICT more than it did in the industry, which was kind of surprising. But yeah, that's how it actually worked out because here at ICT, we were part of a lot of workshops. We organized a lot of them ourselves. And then I also, as being a PhD student, I also had to go and present my work in other conferences. So all in all, I feel like I actually built a larger network while doing my PhD at ICT.
If I had to ask you about the differences between, working in the industry and working at ICT, which is an academic institution, what would you say are some of the differences that you have observed with respect to work culture or networking or the environment?
Yeah, there are a lot of differences actually. I think the most obvious one would be that the industrial environment is more regulated. I mean, it's required to be regulated. So you're supposed to like follow things as per protocol, as per regulatory guidelines, which is very essential. So in academia, we don't really have those requirements. We can if we want to, but they're not necessary. But I think the best part about academia as compared to industry would be you have more freedom to conduct any kind of research that you want. You have the scope of going a little beyond what is required. Whereas I think industry is maybe not always, but maybe a little limited in that case that you have to stick to protocols, you have to stick to your objectives and things like that. So I think academia has more freedom and industry is a bit more regulated, which is I understand the reason for it, but that's just mainly the difference.
What are some things that you like about working in the industry?
I think at a personal level it would be there were fixed days and working hours and things like that. In academia, especially since it was a PhD, it was very intensive. So worked a lot, worked very long days, even weekends, very few holidays. So that way, I think industry allowed us to have a little more of free time for ourselves. And it also gives you a lot of stability in your life because you get a fixed income.
Hmm.
And so you're less worried about how to manage your finances and things like that.
Your PhD at ICT took around 5 years to complete, is it?
Yeah, 5.5. Yeah.
Oh, wow, 5.5 years. What are some of the most memorable moments of your PhD journey that you would like to look back on?
I think the most memorable was when my first publication got accepted because I was struggling with that for a very, very long time. And it actually got accepted just last year in November, which is basically after I finished 5 years. So it was very stressful that I was not able to get that published. And when it finally did, I felt this major relief that finally something is out. And immediately after that, like within a month or two, the second one got published as well. So they were like two big successes for me. But when the first one got accepted, I was extremely excited and relieved. Yeah, that would be the best moment for me.
Sounds really amazing. Now that you're done with writing your thesis, how are you preparing for your defense?
Basically, I'm just trying to read up on things that I was reading a lot, but just refreshing my memory because I've kind of, it's been a few months now since I've stopped working in the lab. So just refreshing my memory and also going more deep into, the reasons for why things worked out the way they did, just to be kind of like, you know, so that I'm able to answer all questions that come up in the defense. So yeah, just trying to make my foundations more stronger and hopefully I can get my defense done well in that way.
Are you nervous for it?
Yeah, very, because I mean, it is the main part of your entire project, and you obviously want to feel confident and show that you have been not only worked enough, but you also understand your project enough. You know how to defend yourself and your explanations and the work, the results you've got. So there is a lot of pressure. So yeah, a little nervous, but also at the same time, like I said, I'm still very, very relieved that everything is completed and I'm at the very end. So it's kind of like a mixture of nervousness and also relief. Yeah.
That's nice. I do have a final question for you, which is if there is anything that you could say to your younger self before they started their PhD, what would you say?
How young are you talking about?
Okay, like what would you say to someone who is just starting their PhD?
So I think if you're in that stage of the initial stages of your PhD, I think more than anything you need is the will to like get to like explore your topic as much as possible, like read as much as possible. So we say that novelty is a very important aspect in a PhD project, but then just the whole thing is also more important, should be more important to you personally in the at the level where you have to learn something new every time you like possibly every time you perform an experiment. So it's also a great learning process. So I think it's very important to keep your like have a strong foundation. You have to read a lot. It not only allows you to be up to date with the work that is being done in your field, but you need to have a strong foundation in order to progress in your work. So if you're in that very very initial stages, you have to make sure that you have designed your project in such a way that is not only achievable in one sense, but you have to be able to understand clearly what you need to do in the next 5 years, 4 or 5 years. So you need to lay out a proper plan, and yeah, just read and work as much as possible to achieve those objectives. And like I mean, maybe as time passes, more than reading and working, your priorities tend to shift to just having patience, because a lot of times there'll be a lot of instances where things just don't work out. So you have to have a lot of patience as well to like just continue trying until it does, and eventually you will get to the end.
Marianne, I really, really enjoyed talking to you, and thank you so much for taking time out of your day to speak with me. I hope the people who listen to the podcast feel inspired by you and your journey and hopefully come and do their PhDs with NRG. So, yeah,
Thanks a lot. It was nice talking to you too.
thank you. And I hope you have a nice day. All the best for your defense.
Thanks a lot. Have a nice day as well. And all the
You
best
too.
to everyone out there who's also planning to pursue a PhD. I hope you all succeed in your endeavors.